So yesterday we were talking about the T cell and B cell differentiation and how they develop not only in terms of tolerance induction so that they don't attack our own cells, but also in terms of having the specificity in terms of their T cell receptor and the B cell receptor to recognize anywhere in the neighborhood of 10 to the 10, 10 to the 11 different type of antigens. And today what we're going to talk about is antigen presentation and how those innate cells present to, or even B cells present to the T cells in order to express adaptive immunity. So remember we talked about the antigen presenting cells, and just to review that, you've got your macrophages, your dendritic cells, and then also your B cells. All right? So these are going to encounter antigen out in the system, a pathogen if you will, and then they're going to present that to a T cell. So let's take the dendritic cell for example. And I think this is, as we talked about, one of the most remarkable cells in the human body. So it's a beautiful cell. It kind of has these star-shaped like fingers, if you will. And the dendritic cell, to give you an example, let's take the GI system. Okay, so you have the lumen of the gut, and your immune cells are not privy to the lumen of the gut, because if they were, they would get really excited seeing all the kind of different pathogens and different antigens that you might have in the gut. And so the dendritic cell is very unique in that, let's say that this is the lumen of the gut and this is your tissue, and the dendritic cell reaches his little process up into here and he samples to see if there's any particular bad guys lurking around trying to invade the mucosal of your gut. Well, he processes that antigen, and if in fact it is a pathogen, he starts to process that like we talked about in terms of the proteasome, cutting up the guts of that, displaying it in his MHC class II molecule. Now that dendritic cell is able to, very unique cell, in that it's able to take the fight, almost like he puts the bad guy in his cop car, and now he drives to the police station. And the police station is going to be that secondary lymphoid tissue, okay? So that could be your lymph nodes, that could be your mucosal-associated lymphoid tissue, the spleen, for example, perhaps, but let's focus on the lymph nodes. And so where that is, that secondary lymphoid tissue, is where the T cells are hanging out. And one example that I heard that was really helpful was from an MIT podcast on OpenCourseWare, and the way that it is expressed is this dendritic cell lives to present his antigen to the T cell. And when he goes by, there's all these T cells, and remember the T cells have specific receptors, as we talked about the differentiation yesterday. Now, as that comparison is, essentially, it's as though the dendritic cell is driving by this souk, essentially, of all these different little, if you've ever been to the Middle East, they have all these different wonderful little shops, and he's trying to shop his antigen to these T cells. Well, lo and behold, one of them recognizes what's in the pocket of that MHC class II molecule is also going to be comparable to the specific receptor for the T cell. So let's say that you have this antigen presenting cell. He's got a bad guy sitting in his MHC class II molecule, and remember we can have several of MHC class II molecules because we're all talking about chemistry here. And then he's got his little girlfriend, the T cell, okay, and she has, so this is your MHC class II molecule, and this is your CD4 positive. Remember, 4 is being presented to MHC class II. And this is going to be the reaction with the T cell receptor, okay? So that's essentially the first chemical reaction, if you will. And one example I read in a textbook was that it's as though you're starting the car, you're starting the ignition. But that signal is not enough to activate this T cell, okay? So you have to have a second signal, and they call that co-stimulation. And in this case, the T cell has what they call a CD28 marker, cluster designation 28, and that co-stimulatory molecule on the B cell is your CD80, sometimes 86 they call it, and even to be more confusion they'll call it B7-1 or 2. So all these names essentially mean the same thing, and it's the co-stimulatory molecule. So we started the ignition with the relationship between MHC class 2 and the T cell receptor, and now we're going to put that car in drive with the co-stimulation. Now there's some other intramolecular reactions that we'll go into a little bit later, but these are the two probably most critically tested, if you will, of this interaction. Now, once this happens, the T cell begins to secrete cytokines. Remember that cytokines are like the growth factors of the immune system, okay? And they're also, you can call them like hormones of the immune system. So it starts to secrete interleukin-2, all right? Now that is going to cause an increase in the proliferation of this particular T cell. So now you start to get a monoclonal proliferation, a growth factor, if you will, of these T cells. Now, not all T cells behave the same way. There's what they call your Th1 T cells and your Th2 type cytokines. And so that's what we're going to go into now. And this is going to be, again, one of the most critically tested areas. So we talked about antigen presentation and how the T cell is activated by that antigen presenting cell. Remember that macrophages can do that and B cells can do that as well. Okay, so now let's step back and have the background of what we just discussed in mind, okay? And so we start off over here, and this is a CD4 positive T cell, and let's call it a TP cell, essentially, okay? So this is the T sub P, all right? And then that's going to differentiate into another T cell, and we call this a T sub naught cell, okay? So I think PO, you know, you can come up with whatever acronym that you want. And then, depending on the hormones, if you will, the cytokine hormones, the cytokine milieu, as some people say, then this T naught cell will differentiate into a Th1 type T cell in terms of secreting cytokines, or a Th2 type cytokines. Now, maybe that doesn't make sense, but it will in just a minute, okay? So the first thing that happens is this, you've got an antigen presentation here, okay? And you start to secrete interleukin-2. Now, that, again, is a growth factor for T cells. There's drugs that target interleukin-2. So cyclosporine, for example, targets that. And so you would shut that down, and then you wouldn't get all this reactive T cell activity. So that's why it's important to know these, again, because you've got drug targets associated with them. Okay, so that's going to have IL-2. And then subsequently, like we talked about yesterday, you have this CD25, which is nothing more than an IL-2 receptor. So that essentially feeds back. So it's sort of an autocrine type of a situation, and you get monoclonal proliferation of these T cells. So you can see why IL-2 is a very critical, one of the first discovered T cells, okay? And, or one of the first discovered cytokines. So IL-2, all right, is going to increase the proliferation of the T naught cells. And these guys, again, we're talking about CD4 positive T cells, alpha beta T cells. This is going to, is kind of neutral. In other words, I call it the Switzerland of T cells, okay? So this is going to secrete IL-2, it's going to secrete IL-4, and interferon gamma, okay? So essentially, the reason why it'll make sense in a second why I call it the Switzerland, because it's sort of neutral, as I mentioned. So the guys that are going to increase the expression of Th1, okay, that's going to be your, essentially your IL-2 and your interferon gamma, okay? So, and then what's going to increase the expression of Th2 is your IL-4, right? So the way these cells behave is very different in terms of the Th1 cytokines. This is what I call the George Bush cytokines, George Bush II, okay? It's sort of a bit of a kill 'em all, let God sort 'em out kind of cytokine in terms of you're going to have all your firepower directed towards that pathogen, all right? And the reason is we talked about before, remember, interferon gamma increases the expression of MHC class II molecules, so you can keep presenting more and more and say, look, at the bad guys are here, the bad guys are here, so you can continue to feed this fire, if you will. Interferon gamma is going to increase the expression of NADPH oxidase. Remember, that's that reactive oxygen species. It's going to increase the expression of INOS, which is also inducible nitric oxide, and that's also very toxic to bad guys. And it's also going to essentially allow you to neutralize intracellular pathogens, like, for example, Mycobacterium tuberculosis. TB is quite an interesting little tricky guy, and you will form what they call a granuloma, which we'll talk about when we get to more into the micro aspect of immunology. So it's very critical. In fact, a certain type of leprosy, okay, lepromatous leprosy, if you don't have Th1 cytokines, you will not be able to clear that, as opposed to if you do, you can clear that type of leprosy. If you have instead the Th2 cytokines, then you're going to have a perpetual form of leprosy, which again we'll talk about. So that's where, in terms of immunology and the link with microbiology, is you want to be aware of these cytokines, why they're so important. Okay, now, so these are essentially, this is what perpetuates the Th1 cytokines. So like I talked about, this is going to give you an IL-2, it's going to give you an interferon gamma, and tumor necrosis factor. Tumor necrosis factor we'll talk about more when we get to inflammation, but again, make a note of the fact that tumor necrosis factor is one of, an anti-tumor necrosis factor is one of the largest selling drugs on the market today, okay? Many different varieties of that particular target for TNF. This will kill you if you got too much. They call it septic shock. Okay, so that's again why you want to think about these cytokines. Now, in terms of the Th2 cytokines, this is a long list, all right, but let's go through it. So you've got the type of cytokines that it's going to secrete would be IL-4, IL-5, IL-10, IL-13, Tgf-Beta, okay? So the way that I sort of think about this is, again, you know, I think of it's a, you know, 10-4, good buddy, thank God it's Friday the 13th. Oh, by the way, we'll have a six-pack. Okay, so these are all IL, and that's an IL. Okay, so let's talk quickly about what these different cytokines do. Again, this is the heart of immunology, all right? So IL-4, you need that to what they call isotype switch to IgE. Remember, we talked about IgE with regard to allergy. We'll talk about it a lot more, and also with regard to killing parasites, particularly helminths, all right? IL-5, that is going to be secreted by mast cells also, but it's secreted by Th2. And what does IL-5 do? Remember, that was a growth factor for eosinophils. Eosinophils you often see in allergy, and you also see eosinophils are critical to kill parasites, right? With regard to major basic protein being secreted, as well as eosinophil cationic protein. IL-10, oof, this is huge. If you want, this is the antithesis of the George Bush cytokine, okay? IL-10, I call it sort of the party pooper cytokine, because what it does is, remember how we talked about our NK cells out here? All right, so the NK cells are a unique kind of a T cell, if you will. They're of that lymphocyte. It's a non-traditional T cell. So what the NK cells do is they actually secrete interferon gamma, okay? And the way that they do that is a friendly macrophage secretes IL-12 when he gets activated. So he recognizes a bad guy, he processes it, he starts to secrete IL-12, and also in the presence of IL-18. If we remember that from way back, the inflammasome was created in order to secrete IL-18 or activate IL-18, or also in the presence of IL-15. So these guys basically enhance the sort of power of that. It's almost like a mixed drink, if you will, for this NK cell, right? Instead of one flavor of alcohol, he's got two, and he gets very, very loosened up here, and he starts secreting interferon gamma. And looky here, interferon gamma then drives the Th1 fight, okay? Now, where was I going with IL-10? All right, that's a long way of saying that IL-10 actually reduces the secretion of IL-12. So you can see IL-12 is critical to get the NK cell to be able to secrete interferon gamma. So it essentially suppresses the Th1 response. And that's why, you know, I call it the sort of party pooper cytokine, if you will. IL-13 is going to be something we'll talk about more when we talk about allergy, but just put that sort of in the back of your head. Tgf-Beta. Now, Tgf-Beta, there's many different applications of it, but in terms of where we're talking here, it downregulates your ionos. It's going to downregulate some of your reactive oxygen species that we talked about with regard to the macrophages and their ability to clear pathogens. So that's one thing. It does a lot of other things as well, which we'll get into. IL-6. IL-6 is essentially a growth factor of what we call our acute phase type reactants or proteins. So IL-6 we'll get to when we talk about inflammation, but essentially what IL-6 does is it enables the vessels to become, to be able to attract and bring in your immune cells, all right? So we'll talk more about IL-6 because that's going to increase the expression of C-reactive protein, which is part of opsonization, and it's also going to increase the expression of the adhesion molecules that allow these cells, these immune cells, as they're driving down the street, to be able to stop and enter into the tissue where there's a particular pathogen or where there's tissue damage. So that's what the role of IL-6 is. So again, this is probably, in terms of testing, is probably one of the most important things that you can get your mind around. What is a Th1 cytokine? What is a Th2 cytokine? And what drives them? IL-4 here, interferon gamma is what's driving the Th1. The only other thing that I'll mention is IL-2 is also very instrumental, okay, in turning this NK cell into what they call a LAC, which is a activated killer. I think it's called lymphocyte or lymphokine. Can't remember. Activated killer. So it just makes it an even stronger killer than normal. All right. And then the other aspect of it is that IL-2 will push not only a CD4 differentiation and proliferation, if you will, but it will also increase the expression of CD8 positive T cells. All right, and remember, those are your cytotoxic T cells. So those recognize the MHC class I molecules, which is increased, right, in the expression by interferon gamma and also interferon alpha and interferon beta. And then, looky here, this guy comes in and is able to eliminate those cells because, again, he's got the ability to lyse the cells with perforins. All right, so let's go to some questions now on this section. So question number 1 is, what cytokine do dendritic cells secrete that activate NK cells? And the answer is E, IL-12, right? See this guy right here activates the NK cell. And then number 2 is, what does IL-12 do? And the answer there is C, it activates NK cells to secrete interferon gamma, okay? Number 3 is, what is interferon gamma's role in the differentiation of T cells? Well, we know that. It's B. It pushes that Th0 cell, all right? Here's the P, here's the naught. It pushes the Th0 cell over here to the Th1. And really, it's interferon gamma that's pushing this, okay? We can, you can see that you get an expression of IL-2 in the milieu anyway, but it's really the interferon gamma that's pushing that. Number 4 is, what does IL-2 do with respect to T cell differentiation? And that is, C is, it pushes the P0 cell here to basically start into the T sub 0 or T sub 0 cell. Number 5 is so that answer, sorry, would be C. What cytokines do T0 secrete? Okay, and again, it's all of them. It's IL-2, 4, and interferon gamma. From this stage, you can go either way. You can go to Th1 or you can go to Th2. And then number 6 is, what is the cluster designation for IL-2R receptor found on all T cells? And remember, that was over here. That's CD25, okay? So it's just essentially another name for a T cell receptor. And that's on all the T cells, even the regulatory T cells. Number 7 is, what is IL-4's role in the differentiation of T cells? And again, that's just B. It pushes this T0 into your Th2 cytokines, all right? So if you have more IL-4 than interferon gamma, you're going to come onto the Th2 cytokines. Number 8 is, what cytokines do Th2 cells secrete? Okay, so we talked about those. That's going to be essentially all of the above. So you got, 10-4, good buddy, thank God it's Friday the 13th, and we've got 5, and we're going to have a 6-pack. So all of the above would be G. Number 9 is, what cytokines do Th1 cells secrete? All right, so we love these. This is your IL-2, your interferon gamma, and your TNF. Number 10 is, what does IL-10 do with respect to regulation of T cells? So it's an indirect regulation of T cells. And remember, IL-10 here is the party pooper because it reduces the secretion of IL-12. Without IL-12, you can't push these cytokines. You can't push, the NK cells to be able to secrete interferon gamma, which is critical to differentiation of Th1. Number 11 is, which of the following is the T cell regulatory mechanism at the level of the antigen presenting cell? So we talked about this again yesterday, but let me go through, let me fully go through it. So remember that when we had the presentation, so just bear with me, and you've got a T cell receptor, okay, and this is your CD4 positive cell, and you have a macrophage with your MHC class 2. We talked about first start the CAR MHC class 2 with the CD4 receptor, okay, and then we talked about the co-stimulation with CD28 here and your CD8086/B7-12. Well, guess what? There's also, instead of putting it in drive with co-stimulation, you can put it in reverse, all right? And in this case, you still have the macrophage with the same cluster designation, CD8086, but the T cell has a CD152 on the T cell, all right, or they call it CTLA4. So as you can see, is the immune system will activate, but it has these intrinsic mechanisms by which it can downregulate as well. We talked about the other mechanism of that, which I'll review in a second, but the answer there is, on number 11 is B, all right? So we talked about that. You also have AI Tregs, you have N-Tregs from the thymus, and remember, you can also shut down that little macrophage, right? He's got a CD200 receptor, and a T cell or some other immune cells have a CD200, okay? Binds to the receptor, remember retard, and this guy can no longer secrete the kind of cytokines, these cytokines that drive this process. So there's many different regulatory processes that hopefully work, otherwise you have immunopathology, which we'll come to as well. Number 12 is, which type of T cells drive cytotoxic CD8 positive differentiation, all right? And that's going to be your Th1 cytokines, because again, this IL-2 is going to drive that process, okay? So that's who's going to drive that. So the answer would be B. Number 13, what does the cytotoxic CD8 positive T cell interact with? And we've talked about that. That's the MHC class 1 molecule, MHC class 1. all right, displayed on all nucleated cells. Number 14, which cytokines cause clonal expansion of the PT cell, all right? And so the answer there is going to be D, which is interferon gamma as well as IL-2. Now, the last bit of this, let's talk briefly about a different kind of cytokines. So we talked about Th1 and Th2 cytokines. And remember, as I'm erasing this, in immunology, it's constantly changing due to the research that's coming out. In fact, I had bought this old review book, question book, written about 10 years ago. It was like used, and it was about $2, I think, not even, at the library. And half the questions in there were wrong because we've learned so much, okay? And Th17 cytokines are a part of that, learning process. So we talked about Th1 and Th2 cytokines, but there's also another type, well, there's many types, but Th17 cytokines is now an area of a great deal of research. And when you think of Th17, you want to think of inflammation in general. And the way that you get a Th17 cytokine is Tgf-Beta is what proliferates or causes the proliferation. And it can be with IL-1 or IL-6, okay? And also in the presence of IL-23. Okay, and IL-23 is secreted by dendritic cells. Is who secretes the IL-23. So this is just another sort of section, and we'll talk about it with a couple of review questions. Here is number 1 on Th17 cytokines is, which of the following cytokines cause Th0 cytokines to differentiate into Th17 T cells? And the answer is A, Tgf-Beta plus IL-1 or IL-6 and IL-23. Again, this is a dendritic cell secreting that IL-23. Number 2 on this is, what is the role of IL-23? And it's secreted by activated dendritic cells with respect to Th17 cytokines. And the first answer is correct. It stabilizes the Th17 cells, and they in turn secrete IL-17, A, F, and IL-21 and 22. Of course, the next question is, which of the following are true with respect to interleukin 17 and interleukin 17F? And this is a laundry list of things that these cells do, and you can understand why they're very instrumental in inflammation. All of the above are true, but let's just go through it for the sake of the fun that we're having with inflammation. Is A, they induce pro-inflammatory cytokines, which we talked about. They induce metalloproteases. Now, metalloproteases are sometimes put into this acronym, MMP, as I recall. Metalloproteases, what they do is they enable you to drill through the collagen type 4, which remember is the basement membrane, what the basement membrane of a cell is made up of. So guess who obviously loves this metalloprotease would be your cancer cells, and that's how they in fact invade, or one aspect of how they invade and metastasize. But it's also a function of your immune cells to be able to get to the fight is through metalloproteases. So these IL-17 as well as the Th17 cytokines is what induces that. C, they induce chemokines. So they are in fact chemotactic. In D, IL-17A is chemotactic for neutrophils. And then E, they play a role in fungal infections. So all of the above in terms of Th17. Thank you.